Free transcriptome assembly of Nanopore RNA-seq data.
Hi, everyone. My name is Ming Nip. I'm a PhD student from Dr. Inashborough lab at the BC Cancer Genome Sciences Center. Today, I'll talk about a method that I have developed for reference-free transcriptome assembly of Nanopore RNA-seq data. Nanopore RNA sequencing data are known for having very long reads, but these reads have a very high error rate, and because of this, the reads are usually corrected for errors before analysis. Although the reads may capture full-length isoforms, the long transcript isoforms would require assembly. The current state-of-the-art reference-free assembly methods are mainly designed for a hybrid of long reads and short reads. So here we present our method that's intended for reference-free assembly of purely Nanopore RNA-seq data. Our method is implemented in a program called RNA Bloom. Our method consists of 5 stages, and it assembles isoforms from raw nanopore reads. So I'll briefly go over each of these 5 stages. In the first stage, we constructed the burn graph from the reads, and to do this, we estimate the cardinality of the k-mers in the reads using a tool called NT-card. So basically, NT-card will generate the k-mer coverage histograms as shown here. And from this histogram, we are able to identify a threshold in k-mer coverage that distinguishes between weak k-mers and solid k-mers. So basically, weak k-mers are saturated with errors, whereas solid k-mers are mostly free of errors. After that, we extract all the k-mers from the reads, and we store them in a memory-efficient data structure called Bloom filters. So using the burn graph constructed from the previous step, we could perform alignment-free error correction of the nanopore reads. But before we do any error correction, we could discard all the poor quality reads. So these would be reads that are either too short or they have a high proportion of weak k-mers. And for the good quality reads that remain, we perform error correction. To correct the errors in the reads, first, we trim the weak k-mers from both ends of the read, and then after that, we try to replace all internal weak k-mers with solid k-mers. And this is done while keeping high % sequence identity. So after the reads have been corrected for errors, we stratify the reads according to the read length and median k-mer coverage. So the reason for this stratification is to prioritize the processing of long and high coverage reads before short and low coverage reads. After that, we try to cluster the reads belonging to the same gene using minimizer sketches. To generate a minimizer sketch from a read, basically, we just need to extract all the hash value from the k-mers in the read. And for a sliding window of a fixed size, we determine the hash value, and this is called the minimizer. The collection of minimizers is called a minimizer sketch. So essentially, the minimizer sketch is a sparse representation of the read sequence. The minimizer sketch is used to represent the reads assigned to the cluster. So if a read matches a sufficient proportion of the sketch, that read is assigned to the sketch's associated cluster. After all the reads have been clustered, we could assemble the reads in each cluster separately. And this is done in an overlap layout consensus manner, and this is often seen in many reference-free genome assembly algorithms. So in RNA Bloom, the overlap step, so this is the overlap of the nanopore reads, it's done using Minimap2, and the layout of the overlap reads is generated with Mini-ASM. And finally, the consensus of the sequences is generated with Minimap2 and RADCON. So essentially, this is basically just a genome assembly workflow with some adjusted parameters. So to evaluate our method, we generated a simulated data set with trans-nanoSym. So basically, trans-nanoSym will extract the error profile, read distribution, and the expression profile of the isoforms, and will generate nanopore reads from the most reference isoforms. And in our data set, we have roughly around 440,000 reads, and the read length ranges from 36 bases to 230,000 bases. So first, we evaluate the error correction algorithm in RNA-Bloom. And as shown in this figure, we can see that RNA-Bloom is able to reduce the error rate from over 8% down to below 3%. Next, we look at the accuracy of the clustering algorithm in RNA-Bloom. And from these figures, we can see that RNA-Bloom is doing a pretty good job in clustering the reads into genes. So the chart on the left shows that 96% of the cluster are single gene clusters. And the chart on the right shows that 86% of the genes simulated are assigned to single clusters. And finally, we evaluate the quality of the assembly of RNA-Bloom, and we compare RNA-Bloom against two methods: FLAIR, which is a reference-based transcriptome assembler, and Mini-ASM. So this is basically the overlap layout consensus workflow that I have described earlier. And we assess the assembly quality using RNA-Cross. So here is a subset of the RNA-Cross results. And we can see that RNA-Bloom has the best transcript reconstruction compared to Mini-ASM and FLAIR. And this is actually surprising to me because given that FLAIR is a reference-based assembly approach, I would actually expect it to have the best transcript reconstruction. What's not surprising in this set of results is that FLAIR has no misassemblies. So in summary, I have developed a reference-free assembly method for Nanopore RNA-seq data. So I would like to thank my colleagues, Saba, Chen, Renee, and my supervisor, Dr. Inash Burrow. And my project is funded by Genome BC, Genome Canada, and NIH. And my travel is funded by the John Baudet Memorial Fund. And once again, my name is Ming Nip. You can contact me at this email address, and you can download RNA-Bloom at this GitHub repo. So today I'll tell you about a method that assembles transcript isoform from Nanopore RNA-seq data, but RNA-Bloom could also be used for Illumina bulk RNA-seq data and also single-cell RNA-seq data, which I have actually given a talk about this last year in ISMB. And here is a list of posters that you could feel free to check out. I think they would need to be taken down by 2 p.m. And I'd like to thank you all for staying here and hearing my talk, and I'll be happy to answer any questions. Thank you.
Hi, very, very nice work. I'm curious about your sensitivity for being able to detect alternatively spliced isoforms, given that you're clustering transcripts, or clustering reads rather, based on the minimizer hashing scheme. And my worry is that you're going to get alternative splicing isoforms, are going to get clustered together, and then you're going to end up with a consensus transcript that's going to represent maybe one of the isoforms or some chimera between the isoforms rather than the independent separate isoforms.
Yeah, that's actually a very good point. So if we look at this table, so if we actually perform that overlap layout consensus step using the default parameters, and like you said, all the alternative splicing events would actually be collapsed together. So I actually would have to adjust the parameters in Mini-ASM to make sure that it will generate the layout for the different isoforms.
Good. The first question, is this published? Do you have a preprint?
This is not published, but I have a paper on bioRxiv for single-cell transcriptome assembly, and that's for Illumina short reads.
All right, okay. So, and another question is, how do you identify those weak chimeras? Like, if you need to know if those weak chimeras are the chimeras contain errors, right? You have to compare to something to know they contain errors. So how do you know they are?
Yeah, so that's actually a good question. So this is actually purely done by looking at the K-mer coverage histogram here, and basically I would set a threshold that distinguish between the two bars in the histogram. So if the ratio falls, or if the ratio falls below a certain threshold, then I would set the threshold at that point on the histogram. So this is just a proxy to distinguish between weak K-mers and solid K-mers. So even though it's called weak K-mers, it's not like, well, like
so
it's not
that-
completely contain error K-mers.
So that means just because these K-mers mapping to multiple places, it doesn't mean that they actually contain errors, isn't it?
No, the weak K-mers are only intended as a candidate for error correction. So,
Okay.
so it doesn't mean that they will always be replaced in the error correction algorithm. It's just candidates for error correction.
Okay.
Last short question.
Yeah, thank you for your talk. Very short question. What is the K-mer size here?
I use 17.
17?
It's my magic number.
Oh, like you.
Well, thank you to all the speakers in this session.